Good morning, everyone. Title of my talk is protein folding and design and for making mechanically robust protein-based materials. So when I saw the poster, this basically it's given importance to proteins because its AlphaFold is actually became very well known in the last couple of years because of the protein folding. Why proteins are actually important? Proteins are beautiful and essential molecules in living systems, and they are important for multiple functions, like in neuroscience, like UP mentioned, or Shruti mentioned in other cases, and also sometimes even catalyzing reactions other than RNA. They come actually in multiple varieties. And what are these proteins? These are nothing but polymers of amino acids. And these amino acids have actually different flavors. So they come in charged, uncharged, polar, hydrophobic, hydrogen bonding, etc., etc. But what's actually important state of these proteins is actually the folded state, where these biopolymers, which don't have any structure to begin with and fold into a well-defined state in most of the cases. In some cases, they are like IDPs, like Shruti mentioned. But in most of the cases, they actually fold into well-defined structure called native state, and this is functional. So if these proteins don't really fold into these states, and then sometimes they misfold and then they aggregate, and then they create actually problems. So in some cases, these aggregates can be cleared because of other proteins inside the cells, or sometimes these proteins actually aggregates and they can't be cleared, and then they create a disease state, some like in Alzheimer's and Parkinson, etc. So AlphaFold is actually became well known for predicting structures from the purely based on the amino acid sequence, because there are a lot of structures available in the PDB. So AlphaFold actually learns the structures of all those existing proteins. And then if you submit a sequence, then it tries to find out the structure and actually does very good job. But what is not really known is actually how this unfolded chain actually goes into folded state. What are the intermediate steps? Okay, what are the sequential pathways in which these proteins actually get into this structure is still not known. It's still unknown, unsolved problem called protein folding. So today I'm going to talk about one specific function of the proteins among many different functions. Is basically function which is associated with stretching force or mechanical force. So if you see these two pictures here, so the one is both are taken from the natural world. So one is basically muscle tissue in us, and another is basically spider silk. So at the prima facie, they look actually completely different from each other, but there are actually some commonalities between these two systems. Is that both are actually mainly made up of protein molecules, and then these are actually have ultimate strength. So if you see the spider silk, if you see these strands, they have actually thinner than human hair, roughly 3 orders of magnitude smaller or 1,000 times smaller. But they are actually so strong that they're actually stronger than steel, okay, for the same dimensions of steel if you take. But how actually these molecules or this material made up of purely on the protein actually achieves this ultimate mechanical strength is not really known. At the same time, in the case of muscle proteins, so muscle tissue is actually much more complex than spider silk, but yet there are actually some elements which are common between the proteins which actually act as a scaffold in these cases. So in both cases, you can actually treat proteins molecular springs, means that if you know the mechanical property of these molecules, then actually you can model their function. And then main common theme between these two systems is basically the proteins, how actually they are organized in the form of a network. Okay, so the spider silk, okay, again is made up of mostly proteins at the core, and of course it has some sheath outside skin covering basically to act as an adhesive for basically small insects. But if you see the core, it's mainly made up of mostly one type of amino acid, basically alanine, and next predominant amino acid is glycine, and other 18 naturally occurring amino acids are actually very, very rare. Yet actually this attains ultimate strength because of the complex architecture of this protein. So mainly alanine-based sequences actually organize into what are called nanocrystals, and then glycine-based sequences actually have amorphous structures. So both structured and unstructured regions come together and gives rise to this ultimate mechanical strength. If you see the muscle architecture tissue, it's very, very complex. But if you really zoom in into the cell level or into the individual cells called myocytes, you see very well organized structure of protein materials made up of motor proteins, and where actually they slide against certain filaments. But at the same time, there are molecules which actually connect these motor proteins on the filaments to rigid walls through other protein molecules, and which are actually act as passive elements, which their function is to sense forces and then basically limit the extension of this muscle substructure called sarcomere. But so at the core, basically again, protein molecules are involved. Just like in the case of spider silk, where so proteins which are in the structured and unstructured form come together to perform function. So this molecular sensor, which is part of the sarcomere, again has very complex architecture, which is really long polymer chain, longest polymer chain you can really find in the body, made up of structured regions called immunoglobulin regions, where if you see roughly about every 100 amino acids, they have well-defined structure. But however, actually they are interspersed by unstructured regions, which actually don't have any well-defined structures. So basically this architecture is common for all types of tissues. So what do we do in our lab? So to measure the mechanical function of these molecules, either in the spider silk or in the muscle protein titin, so we have to actually work with the individual molecules. So this is where actually I was trying to say this is really throughput. You can really see the one molecule at a time you need to really work with, because there is no way ensemble molecules can be subjected to force simultaneously and then measure its mechanical response. So how do? We actually make meticulously these protein molecules using variety of molecular biology tools, and then subject to single molecule experiments where you can really hold single molecules and then manipulate them using force-based tools and then measure the mechanical response. So why it is important? Because you need to really understand at the molecular level how the force response is actually coming or emerging at the entire molecule or even the entire substructure and entire the tissue cell like myocyte. But what is not really known is that how exactly these molecules, what kind of forces they are subjected to inside the cell. So what is their mechanical response? Can we really understand this in the laboratory setting? So second thing is that, so like I said, this muscle cell itself, the titin molecule itself is so complex that it has many different types of domains which vary actually in sequence. So we need to really understand how the variance in the sequence of these molecules gives rise to a similar structure, yet different mechanical properties. Then we need to really understand the correlation between basically protein structure, and if you introduce any mutations, how exactly it gives rise to basically kind of disease models, because there are mutations in the titin protein which actually cause certain diseases like muscular dystrophy and etc. And this problem is actually multi-scale because you need to really measure the properties at the molecular level, and then you need to really understand the whole muscle cell or the muscle tissue. So it basically scales range from molecular scale like few nanometers to basically few microns, and if you take the tissue, maybe even hundreds of microns. But what are the limitations? This is really the low throughput tool, because you have to really work with molecules. You can't really use ensemble of molecules. And the problem is again difficult because it's basically applying force to these structures gives rise to different properties depending on how the force is actually transmitted through this molecule. But through rational design and understanding the current understanding of the mechanical properties of certain elements, people actually have started building more complex architectures where you can take protein molecules and then add on components where binding and force response you can bring into, and then make materials with resilient structures. And by varying the components in the solution, you can actually change the mechanical strength of these molecules. So we want to really make these protein-based self-repair materials, because these materials, because they are protein-based. So for example, let's say if you make a crack here, you can actually repair through protein-protein interactions. And if we make actually proteins with tailor-made mechanical properties, they might be even applicable in biomedical applications. So I'm not really a ML or a DL person, but I would think that this problem requires ML and DL inputs, where we can actually have information which is existing from many different proteins. This is here, the number is just 100. So very small data set, but still we can think that a lot of conditions under which the properties are actually available, measured properties. So maybe we can actually use that data set to even bring in ML and DL to actually understand the problem, and then maybe design based on AlphaFold, maybe new sequences, and then come back and then test their mechanical properties. So with this, thank you, and I'm happy to take if you have any questions.